would ask you please to stand up if you can for the national Moroccan anthem. Thank you very much. Shukran, merci. Again, welcome. Sayyid Wali, Sayyid Rais, Sayyid Mudir, marhaban bikum. Welcome to all of you. And so I know that most of you is coming from so far away and so happy and proud and honored to welcome you in Morocco, in Laayoune. Ladies and gentlemen, unfortunately, the president of the University Mohammed VI Polytechnic couldn't make it with us today, but he insisted on being live with us from Ben Greer on a video. Let's listen to him.
Okay, do you hear me?
Yes, we do.
Okay, Assalamu alaikum, good afternoon, distinguished guests, colleagues and partners, ladies and gentlemen. It's a pleasure to join you virtually for the opening of this third edition of the Laayoune Forum on Biosaline and Arid land agriculture. While I would have preferred to be with you in person, I remain deeply connected to the ambition that brings us together, transforming agriculture in marginal and saline environments from a constraint into an opportunity. Today, over 1.1 billion people worldwide live in arid and semi-arid zones. In Africa alone, more than 60% of the continent's landmass, home to around 200 million people, falls under that classification. Land that is often undervalued but holds untapped potential. At the same time, over 800 million hectares, about 6% of the planet's land, are affected by salinity, with its figure expected to rise due to increasing climate stress, poor irrigation practices, and rising sea levels. This is the context in which our forum takes place, a reality that is both local and global. In Laayoune, in the southern territories of Morocco, we stand at a crossroads. On one side, the pressing need to advance scientific research tailored to harsh, water-scarce environments. On the other, the imperative to turn that research into real-world, scalable solutions that work for farmers in arid and saline regions. In this intersection between frontier science and practical applications, that defines our mission. At UM6P, through ASARI, we have chosen to invest in long-term applied research focused on marginal environments, not as an exception, but as a strategic lens for Africa's food systems transformation. Biosaline and arid land agriculture is no longer a niche. It is test bed for the future of farming across large parts of the continent. This year's program reflects that ambition. It spans salt-tolerant crop breeding, precision mapping of salinity, soil amendments, and beneficial microbe association. Over the next 3 days, the diversity of work displayed here points to one clear truth. Research must not remain confined to the lab. Science on its own is not enough, not if it fails to deliver real solutions for farmers, especially smallholders. What we need as innovations that are field tested, cost-effective, and scalable within local realities. To all the scientists, institutions, students, and development actors contributing to this collective effort, thank you. Your presence in Laayoune, whether physical or virtual, speaks to a shared commitment. We must design new agricultural models tailored to the realities of water scarcity, soil salinity, and climate volatility. Not imported models, but co-developed ones, grounded in science, informed by data, and shaped by local knowledge. Finally, I will to express my gratitude to our co-organizers, particularly the Phosboucraa Foundation, ICARDA, and CGIAR for their continued engagement in this work. I wish you a productive forum and look forward to seeing how the outcomes will feed into practical solutions for African agriculture. Thank you so much.
Thank you very much, Mr. President. Now, ladies and gentlemen, as it is mandatory, we have to show you exactly how we do events, and safety goes first. Ladies and gentlemen, please welcome Lubna to have some words about safety today.
Hello, everyone. I'm Lubna Shafi, Quality Health Safety Environment Manager here at Asari. First of all, I want to welcome you warmly to this big event, LAFOBA 3rd edition, here in the heart of the Sahara. So, as you already know, safety is our top priority. So for that, I will share with you some safety and security instruction to ensure your well-being during this event. Perfect. So in case of fire or emergency alarm, please ensure that each room has the specific fire exit. As you already know, we have 3 rooms, room E, B, and C. Here in room E, we have 2 fire exits, and in room B also we have 2 fire main exits. And also in room C, we have 1 behind you and another 1 in front of you. It depends on your seat. But make sure to use, please, the nearest available exit dependent to your seat. So in each building, of course, we have evacuation plan, and also we have established the extinguisher and all items related to safety and security to ensure your well-being here. And of course, we have evacuation team. So each room, from room E to C, has a team to help you in case of evacuation. And our final destination is assembly points near to parking area. We have 4 assembly points. And for hygiene and facilities, of course, we have washrooms in building D, just in your right-hand side. And of course, hand washing encouraged regularly for your safety. And finally, don't worry, all of our procedures and instruction of safety are under control. We have evacuation team, we have safety and security system here, and enjoy this big event and explore our scientific part here at Asari in the heart of Sahara. And welcome you again to Asari in this big event, LAFOBA 3rd edition. Thank you so much.
Thank you, Lubna. I feel really safe now. I'm good. There we go. Thank you. Again, welcome to Morocco. Welcome to Laayoune. This is a very big event, and I'm really proud and honored to see the list of people that we have here today in Laayoune, names that are expert in the subject of today's. Yes, Mr. Director. Ladies and gentlemen, I would welcome on stage the Wali of the region of Laayoune, Saqa al-Hamra, to welcome you to this beautiful city.
بزاف. الحمد لله وحده، والصلاة والسلام على أشرف المرسلين. أود في البداية أن أرحب بكم جميعا كخبراء وفدوا إلينا من دول أخرى ومن قارات أخرى، وكخبراء مغاربة وكمتدربين معنا في هذا الملتقى الثالث الذي يعتبر من الأهمية بمكان نظرا لأهمية الموضوع، لأن الموضوع مرتبط بطبيعة الحال بالفلاحة، وما يرتبط في الفلاحة هو الأمن الغذائي، والتحدي الأساسي الذي تواجهه الإنسانية هو الغذاء، والغذاء يتأثر بالنمو الديموغرافي خصوصا في قارتنا الإفريقية وفي العديد من دولنا العربية وحتى من دول آسيا، نظرا لشساعة المساحات الأراضي القاحلة والمالحة. فالأهمية أهمية الأبحاث التي تقومون بها كخبراء في مختلف المختبرات وفي جميع المعاهد التي تشتغل في هذا الباب لها أهمية قصوى، لأنه هناك وهم هو أننا يمكننا أن نتحكم في الطبيعة. هذا وهم. الطبيعة لها قوانينها، الطبيعة لها عواملها التي تحكم فيها، ولا يمكن للإنسان أن يتحكم في شيء واحد هو أنشطتنا، هو طرق اشتغالنا سواء في الزراعة أو في الفلاحة أو غيرها. وبالتالي فإن أبحاثكم ستمكننا من ترشيد طرق عملنا، ومن كذلك خلق تقنيات وهي ممكنة لتوسيع الفضاء الذي يمكن استغلاله، لأنه اليوم يمكننا أن نتحكم في الملوحة، يمكننا أن نتحكم في طرق اشتغالنا للتربة، يمكننا أن نبدع في عملنا اليومي، لأنه كما قلت البشرية خصوصا في دول الجنوب لها تحدي أساسي، وهو نقيض التحدي التي تواجهه دول الشمال، هذا التحدي الديموغرافي. هناك بلدان تعاني من نقص النمو الديموغرافي، وهناك دول تعاني. كما تعلمون فإفريقيا ستكون هي ثاني قارة من ناحية الديموغرافية بعد الهند، وهذا نظرا للتحولات والتغيرات المناخية، نظرا لطبيعة التربة، نظرا لطبيعة المناخ، نظرا للأزمات السياسية وغيرها، فإن تحدي الغذاء مطروح اليوم وسيبقى مطروح اليوم غدا. وأكيد أن أبحاثكم المتعلقة بالأراضي القاحلة وبالأراضي التي توجد فيها مياه ولكن مالحة، وكما تعلمون فإننا إذا تمكنا من التحكم في الملوحة نكون قد خلقنا من الماء كل شيء حي. فمتمنياتي لندوتكم هذه بالتوفيق، وليست لي أي خبرة في المجال الفلاحي لكي أتحدث كثيرا، فأنا جئت هنا لأستمع لعروضكم ولأبحاثكم. شكرا ومرحبا بكم في العيون.
شكرا جزيلا سيد ولي على هذا الكلام رائع. Well, ladies and gentlemen, a gentleman that started the project is here with us. He's the chair of this event. Ladies and gentlemen, please welcome Professor Abdelaziz Hirish.
Good afternoon. Ladies and gentlemen, honorable guests, we are all, on behalf of the organizing committee, we are delighted and very happy to welcome you for the third edition of LAFOBA conference. And more specifically, we are happy to welcome you in Laayoune, in a desert environment full of challenges. So I will give just few insights, an overview about LAFOBA, giving some numbers about the conference, just to give you like the context and participant profile, etc. So we started actually thinking about this event and the needs to talk about this problematic that our agriculture here in the region, more specifically, and even in Morocco and other parts of the world are suffering from, which is salinity and drought. And the first edition was held in 2019, even before we started working here at the center. So our research center started in 2020, just before the pandemic. So, and actually, one of the recommendations, from this conference is to develop this program about agriculture in marginal environment. So the first edition, we have just like a little bit more or less 10 presentation with 200 participants. So we are evolving, we are growing, and we are attracting edition after edition interest from many researchers in the world. And we organized in 2022 the second edition of LAFOBA conference, where we received like 130 abstracts. And you can see that this year we are receiving, or we received more than 200 abstracts that will be presented during the 2 coming days. And more than 300 participants are here actually today in this room, or expected to be in the coming days, representing mostly 25 countries that are represented in person. But globally, taking into account the co-authors, so we have like almost 55 countries that are represented from different co-authors organization. So these are the 11 sessions that we have. So we're covering almost all the research thematic of the problematic. So starting from the state of art, to the mapping, to the physiological aspect, to the use of best practices, amendments, microbes, and you can see crop valorization, up to the socio-economic and policy aspects that are also very important. In this slide, I'm showing you the distribution of different papers that we received, and you can see that there are a lot of interest in most of them, but more specifically, physiology, crop response to different stress are mostly dominant, with the cropping practices and the use of amendment, etc. And actually, this is the science. So all about salinity, we need to look at the mechanism, we need to explain, we need to determine how the crops can resist to this kind of stress, and also how we can cope with this major stress. To give you just a few insights about the distribution regarding the presentation type. So we have 125 oral presentation expected in this edition, and we have 17 keynote speech that will be delivered by prominent scientists and researchers present with us today, and almost 35 posters that are displayed in the reception area, just for your interest. In terms of profile of the participant, you can see the graph in the right side. We have almost the same number between professors, researchers, and PhD students. Look at the PhD students, very good ratio actually. So we have almost 70 PhD students who are, participating in this LAFOBA, and this is actually the future of our research. This morning we organized an interactive workshop. So we invited all the PhD students participating at LAFOBA, and they interacted with leading scientists seeking for advice, giving them some recommendation how to do the good science and what they need to avoid in their experimentation. We had really we enjoyed the first, this morning session, and we hope that all students get benefits from this exchange. So this is just like a distribution of different, paper by countries. A lot of them from Morocco, of course, because we are, facing these challenges here in Morocco a lot, and you can see that in terms of research, we are working a lot on the stresses and how we overcome this problem. We have also different, countries that are presented. We got a delegation from Uzbekistan. They are honoring us in this edition because Uzbekistan also is considered one of the most affected countries with the problem of salinity. And finally, I would like just to inform you that we're going to have awards that will be granted to junior researcher, PhD student, postdoc. We have two categories. We have the best oral presentation award, and we have the best poster award, and we're going to have 3 places. So please stay tuned. And for authors and participants who have the poster, please, we're going to have the poster session tomorrow. Please be next to your poster. We're going to have a committee that will evaluate the poster to give the final result during the closing ceremony where we're going to distribute the awards. And finally, I want just to let you know that we have 3 rooms. This is the room A, the big one, and we have room B just next to this one, and we have room C, which is our conference room. So just for your information. So tomorrow, starting from tomorrow, we're going to split in these 3 rooms. Most of you have received the program, and we have another virtual session for people that could not join us today. So we give them also the opportunity to present virtually. But for those who are present here in person, so please just distribute yourself among these 3 rooms. And that's it. So I wish you enjoyable and delighted experience with us. And we hope that this conference bring a lot of benefit to you guys at scientific level and also at personal level. Thank you so much.
Thank you very much, Mr. Hirish, for your welcoming and for your presentation. Ladies and gentlemen, now it's time to start our plenary session number 1. Cutting edges, science towards improving crop resilience to drought and salinity. And we have the honor and privilege to have with us a man from Saudi Arabia, from KAUST, which is the King Abdullah University of Science and Technology. Ladies and gentlemen, please welcome Professor Mark Testa.
Thank you, shukran. Assalamu alaikum. Bonjour. It's a very great pleasure to be here today, and thank you very much to the organizers for inviting me and for the opportunity to share with you some of our work. When you get old and you've been in the business for 40 years, you start to think you should be giving grand speeches with all sorts of generalisms and overviews. And I'm actually not going to do that. I'm wanting to dive straight in because we're still doing a lot of research in our research group. We're still making a lot of advances. And to be honest with you, I'm still excited about what we're doing and some of the opportunities that we have from primary research and, importantly, I would argue crucially, the delivery of that research. So I have 3 basic take-home messages to kick off with, but then don't go home. We'll have some more details, I hope. And so one of the things, of course, that I think many of you will give in the introduction to your own presentations is that salinity is a growing problem. It's a global issue, and it is one in particular affecting irrigated systems where unsustainable use, in particular of groundwater resources, is leading to accumulations of salts in the soil and increasing salinization. Most large aquifers globally that are being used for irrigation are being depleted, so we are mining groundwater resources. And we're mining groundwater resources, therefore it is unsustainable by definition, and we need to be very concerned about this in both the mid and long term. A second message I have is that having worked with wheat in particular, but many crops over the last 40 years, and salinity research trying to improve the salt tolerance of existing crops, I really think that worthy though that work is and contributions though that can make, step changes in salt tolerance may well be more affected more rapidly by the neo-domestication of plants which already have very large levels of salt tolerance. So if you have plants that are native to your region, or even plants you're importing globally, if we're able to, and they're able to grow in, for example, seawater or very, very saline water, we'll be able to domesticate them, increase their utility for us, whether as food, feed, or forage, and that will give us more chances of more rapidly making step changes in tolerance. There's, of course, a third way, where you can do both domestication of wild relatives along with the increase in salinity tolerance of existing crops. And it's this third way that I'm going to be showing you in quite a bit of detail during this presentation, where we've been making quite a lot of research advances. And the final general point I'd like to make is perhaps slightly more controversial, perhaps worthy of discussion over time, but I'd like to just suggest that it may be better for us as humanity and for the planet, especially in irrigated systems such as we have here, all across North Africa, Middle East, West Asia, a lot of South Asia, is to think more about using the precious water that we have to grow higher value crops, in particular fresh fruits and vegetables, which otherwise are often imported with a very high carbon footprint, and it provides an important contribution to local food security, and also addresses another very important need globally, which is not for more starch per se, but for higher nutritional value. I don't know if you all know what the greatest health problem on the planet is, but it's micronutrient deficiencies. We need to be improving the quality of our food. You can do that by enhancing, fortifying staples, but arguably a better way to do this is through increased local production, increased accessibility to the wider population, especially the poorer, large fraction of our population, of food which is of higher nutritional value. And so this is one of the drivers for my own research, having pivoted, if you like, moved from a focus on wheat in particular to the focus on fresh fruits and vegetables and increasing their salinity tolerance. So I'll just give you highlights of two areas of work which we've been studying. I'll skip over that slide. One on quinoa and one on Salicornia, and then I'll give you the primary narrative on tomatoes that we've been focusing on. Now, quinoa is a wonderful, wonderful plant. I see we have Rick Yellen here. We have Didier Bazile, two crucial researchers globally in salinity tolerance. There are many, many other very, very good workers here in this room. I see fantastic posters and abstracts on quinoa, so it's fantastic to see. We've been doing some work to support some foundational work in quinoa, in particular with the genomics of quinoa, in particular with collaboration of the researchers from Brigham Young University. But we're just still doing a little bit more research in quinoa. It is already significantly domesticated, but it does need more work to really try to turn it into a crop which is more globally accessible, in particular for marginal lands. Now look, this is a crop with good drought tolerance, with good salinity tolerance, but it has terrible heat sensitivity. And so it's a plant for the Altiplano of the Andes. So in collaboration with Rick, who is talking, you on the panel? Yes, good. So you can talk to Rick more. He knows a lot more than I do about this. But we have a PhD student who is working on the sensitivity of flowering and the fertilization, pollination and fertilization process in quinoa. And what we find in particular is that if you expose the flowers to high temperatures about 9 days before anthesis, can you imagine the poor students doing this work? The poor student doing this work. But 9 days before anthesis, if you expose the plant to high temperatures, you get a very significant reduction in fertility. So it's actually the time when you have the pollen mother cell formation and meiosis occurring. And so at that stage of flower development, the flower is particularly sensitive to high temperatures. Yet, when you look at the top panel, there's the mysterious abbreviation CB3. This is a plant collected by Rick Yellen many years ago on the shores of the Gulf of Mexico, Gulf of America, call it what you want, ladies and gentlemen. And there it is, more to the point, it is very hot, very saline, and Rick saw this plant, which is a close relative of quinoa. It's called Chenopodium berlanderi. It is tetraploid, but it is quite distantly, it's quite a long way from Chenopodium quinoa evolutionarily, maybe a couple of million years. Don't know. I defer to Rick on this. But that individual, one plant or one small population of plants that Rick collected does not have that sensitivity to heat. This is a result of a lot of work, I should say, from several years in my lab, that graph. And so now, actually Rick did the original crossings. We're getting a mapping population, we're resequencing the lines, and we're trying to identify the molecular genetic basis for that difference in heat tolerance at that crucial stage of plant development. And that's one example of how you can use wild relatives to try to improve the properties of the domesticated or semi-domesticated plants. So another example I'd like to give you is from the middle photograph of this basic slide I've been showing you, which is with Salicornia. Now, Salicornia is an ultimate salt-tolerant plant. It can grow in full seawater. Remarkable. And it's very unusual. I mean, there are a few hundred species of plants, but only a few hundred species of plants can grow really well in full seawater. Mangroves is probably the most spectacular example. It's a whole tree growing in seawater. But these plants aren't too far behind. Salicornia produces an oil-rich seed, and there's many people working on Salicornia. I've seen actually Sissy she will be presenting during this conference as well, doing very good work on Salicornia. And what fascinated me was a more basic academic question. We'd want to domesticate Salicornia, this oil-rich seed. If we can make it a bigger seed, we can give the plants stronger early vigor and so on. Could really make some contributions to oil production, vegetable oil production. But there's also an academic question here, which may have a long-term applied outcome. Salicornia not only is unusual in being able to grow in full seawater, but it's also unusual in that it's part of a small group of plants which are able to accumulate extremely high concentrations of salt in the above ground parts. So it doesn't have leaves, it just has photosynthetic stems. And in those stems, you can get over 1 molar concentration of sodium chloride dissolved in the liquid, in the vacuoles, in the tissues of this plant. Extraordinary, huge concentrations of salt. And so very, very few plants can do this. So the obvious question for me is, what proteins, what ion transport proteins are responsible for enabling this extremely high accumulation of salts in the vacuoles, in the shoots of these plants? So this was a very, very long period of research here, where we're looking at the proteins that were in those vacuolar membranes, looking at the proteins which were induced by salinity stress in these plants, which are induced because the pumping activity is induced by, the salt pumping activity is induced by salinity. And we found very, very beautifully, and we were very great surprise, was that in among the vacuolar membrane proteins was a very well-known transporter called SOS1. It was originally identified in Arabidopsis. It's a sodium proton exchanger. It's pumping sodium, but it was always thought to be in the plasma membrane. So we very excitedly published a paper about this, and we said it was sodium SOS1, this transporter, this sodium pump, it's got neo-localization. We were claiming that the basis for the salinity tolerance, the sodium accumulation of Salicornia, was that this protein, which was normally in the plasma membrane, was in fact localized in this plant into the vacuole. So really nice paper, beautiful work, very exciting. And just like 1 year after, less than 1 year after we published this work, a paper was published in Nature describing that even in useless little plants like Arabidopsis, which are very salt sensitive, the SOS1 protein could actually also be found in the vacuolar membrane. So this claim that we made that it was a neo-localization explaining this salt accumulation was clearly completely wrong. So that paper only lasted for about 1 year in the literature, and it was a lot of work, and we were very proud of it. So we went back to the drawing boards. I wasn't going to take this lying down. This paper, by the way, is very high quality paper, very good work, led by a young woman, Priya Ramakrishna, who's now got a faculty position in Switzerland, University of Lausanne. So we thought we would take this step another further and take some genomics approaches, apply some hardcore molecular biology to see if we could really find out the basis for the sodium accumulation in these Salicornia plants. And we started sequencing lots of genomes. Now, genome sequencing has become massively easier and cheaper now, massively cheaper and easier over the last couple of years. And so we sequenced quite a few different species of Salicornia. And it becomes very complicated very quickly, very disturbingly. So we find that there's lots of chunks of chromosomes are moving around between different chromosomes. We're finding that a lot of the Salicornia, which is supposedly one species, some of them are diploid, some of them are tetraploid. Some are 2 sets of chromosomes, some have 4 sets of chromosomes. So we've got chromosome arm translocations, we've got multiplications of whole genomes, and it starts to become very difficult. So what we did was we started looking at a comparative genomic approach where we were sequencing lots of different Salicornia species, but then also other closely related species which don't accumulate high amounts of sodium in their genomes, to see if we could find different genomes or genome structures which could be related to this high accumulation of sodium in the shoots of Salicornia. Okay, it sort of didn't work. Sort of unsurprisingly in a way, even though we're trying to focus things right down, so we're just looking at close relatives and so on, we end up with a difficult morass, swamp of information in front of us. So we thought we would take a comparative transcriptomic approach and look at the transcripts which are in Salicornia in low salt and high salt, and then compare that with the transcripts in non-Salicornia, non-salt accumulating relatives. And when you start to do that, you end up being able to sort all of these transcripts out, and you find this little group up here, group 1 genes, which are where the genes are being induced by salinity in Salicornia and not being induced by salinity in the non-salt accumulating species. And guess what? It's still over 1,000 genes in that group. So you still get a complete swamp of information. Some of that's going to be very, very interesting information, but it's just one poor slave, PhD student doing this work. He's a very, very good student. So we start looking at what classes of proteins are being encoded by those transcripts, and you end up with a relatively small group of iron transporters, which could be candidate genes. And you start looking through those candidates, and something really quite interesting comes out. SOS1 is there. And SOS1 is being strongly induced in the Salicornia species, multiple species of Salicornia. We're getting high levels of SOS1 expression, and it's being strongly induced by salinity. And when you look in the non-salt accumulating species, the levels of expression of SOS1 are much, much lower, and they're not being induced by salinity. So we think that SOS1 may still be part of the story, but it's going to be much more complex, and it's actually a quantitative thing, not a black and white thing, nothing standing out and slapping you in the face, chunks of DNA missing or chunks of transcripts not being expressed. It's a quantitative difference. And this makes a lot more sense. This makes a lot more sense. And so, of course, what we then do is start to try to identify the basis for that high level of expression of SOS1 and the basis for the sodium expression. Now, I've grayed this out because my student wouldn't let me show it. Because we're analysing the promoter regions of these transporters and doing a comparative promoter analysis now and trying to identify what boxes, little bits of DNA sequence are different in the promoter regions of SOS1 in these two different sets of plants. And why he won't let me show this is because this is like a preliminary analysis. It's got to be done many times over using all sorts of different programs and all sorts of different bases for analyses. But the really interesting thing is this sort of story that I've just told you is possible. This is a student who's just been working for 1 year, all right? You can do this work now quickly and relatively cheaply. It is accessible to a lot more people than it ever used to be. And I can tell you, even if I'm telling you a story which is ultimately still unsatisfactory, it's still a pretty interesting story, and I think it could become very interesting over the next few months, literally few months. Okay, so that's Salicornia. Now I want to tell you about tomatoes and what we're doing, where we're doing both sort of a neo-domestication wild relative story along with a more standard traditional approach. So what we've got here are tomatoes which have variations in salinity tolerance. We're talking about the wild relatives of tomatoes. And what we were wanting to do was get these wild relatives of tomatoes that are salt tolerant and then start to domesticate them or start to introgress the salinity tolerance using traditional breeding. This is my now wife, just married a couple of months ago, Gabi. She did the original crosses many, many years ago between a salt-tolerant Galapagos tomato and a commercial tomato. And she made these intermediate plants and came very excitedly, dragged me into the greenhouse and said, Mark, here, look, we're starting to cross these salt-tolerant Galapagos tomatoes with the commercial tomatoes. Now what do we do? So she did the cross on her own initiative and then came and asked me the question, now what do I do? And my shoulders sag, when you think about this. We all know salinity tolerance, it's complex, it's multigenic, there's all sorts of problems. I didn't want to turn into a tomato breeder per se. I don't have skills, it doesn't play to my strengths. And that's when I had some ideas on how to try to make a shortcut to actually commercially, realistically, trying to exploit this knowledge we have gained through the wild relatives of tomatoes. So there's a big something else here, and that's what I want to tell you about. Now, tomatoes are generally just a moderately salt-tolerant group of plants. They're not that fantastic. And when you look at the wild relatives, there's really not that much spectacularly high salt-tolerant species. There's one up on the far north of Cape York Peninsula, which we identified, growing on the beaches in the tropics, and that's pretty good. But it's pretty distantly related to commercial tomatoes, I tell you. And then there's the ones which are very famously on the Galapagos Islands, which were reported back in the mid-1970s to have extraordinarily high salinity tolerance. So I had a PhD student work in my laboratory, Evelyn Paez. There she is with Bill Gates. Oh, name dropping again. But at any rate, she fed Bill Gates some of our tomatoes and Salicornia, actually, many years ago. And so she was a PhD student. Gabi was a technician in the laboratory. And she was characterizing a lot of these Galapagos tomatoes, but they really were nowhere near as salt tolerant as the original papers suggested, which was very disappointing. But serendipitously, Evelyn identified a Pimpinellifolium accession of tomatoes which had very high salinity tolerance. So along came my next victim, student, called Mitch Morton. He did his PhD with the Pimpinellifolium tomatoes. 326 accessions are in the Tomato Genetics Resource Center in California. And he studied these and found some individuals that had very high performance in these horrendous conditions that we were using, where we were irrigating with really quite brackish water in this hot, dusty, dry environment, which is part of a field station from King Abdulaziz University. Very kindly gave us access to this field station, to this part of their field station. Remember this photograph. It's very, very important for our narrative. So Mitch did his work, published his paper, did all his stuff. That's all good. We published all sorts of different papers. We were sort of sciencing the hell out of this group of tomatoes, and that was all fine. But I wanted to deliver, not just do the science. And so when Gabi came dragged me into the greenhouse with her crosses, what I thought was, rather than try to breed a whole salt-tolerant tomato, which was commercially viable, was instead, which is a decade of work, if not more, what I wanted to do instead, I had the idea of just grafting with these tomatoes instead. Now, you can't graft with the wild relatives. There's often low compatibility. You often get weaker plants, not stronger plants. But if you graft with the F1 hybrids, you can get quite a lot of benefit. So for those of you, do you all know grafting? Yes? You're okay with grafting? Right, okay. So I won't explain the grafting, but suffice to say, it's a very commonly used commercial technique, especially in greenhouse-grown tomatoes. All right? Remember that previous slide where we had those tomatoes in the open fields. Right, now, actually all commercial rootstocks for tomatoes and other species, including a lot of the woody perennials, are actually interspecific F1 hybrids. And these are often, in tomatoes, hybrids between commercial tomato and a wild relative called Solanum habrochaites. It's quite a distant relative. It's moderately hard to get these F1 hybrids growing. And these have been around for many decades now, probably for about 50 years. But when you look at the catalogues, such as Bayer, right, world's biggest seed company, one of the world's biggest seed companies, you look on their catalogue, they've got, I think it's 6 now, but very, very tiny numbers of rootstocks. And Maxifort, which is about 70% of their sales, as of a few years ago at least, when I could get my hands on some data, that's about 40 years old, that hybrid. So it's a very, very sleepy field of research activity. So we thought, let's go in and see what we can do. Let's see if our lines can work. Our lines, remember, where the wild relatives were identified from field experiments in very, very hostile conditions. And so I actually, from a lot of the PhD work, I mined the papers and identified a relatively small number of the Pimpinellifolium accessions for hybridization. Since then, we've done a lot more relatives which have been chosen on the basis of other parameters, in particular disease resistance, and I'll explain more about why I did that later. So we did a lot of different hybridizations, and even those F1 hybrids are able to give you plants which show quite surprisingly good performance in commercial fields. Now, fields. Most of the tomato rootstocks have been developed for greenhouse production because that cost of investment in getting 2 seeds rather than 1, doing the grafting, growing twice as many plants as you are actually going to grow in the greenhouse, all of those things cost money, time, resources, and so it had always been thought that it was only economically viable to do grafting for high-value production systems. But remember, we were doing our work in these hostile open fields for the selection of the wild relatives. And remember that about, or maybe you don't know this, but what I'm telling you is that a very large fraction, most of the tomatoes, probably 98% of tomatoes that are grown are being affected by the environment significantly and going to be affected more with climate change. 62% of the tomatoes that are grown in open fields simply for processing. And then a very large fraction of the rest of the tomatoes are grown in open fields for fresh use and also in very low-tech greenhouses. 6% of tomatoes are grown in low-tech greenhouses where there's very poor climate control. So keep that in mind, and we thought, let's see if we can crack into this enormous market, which is currently completely untouched by the big seed companies like Bayer and Syngenta. So we go out, do some field trials, spectacular results, huge. I'll show you some data in a few minutes. But this is the very first field trial that we did in northern Egypt in collaboration with Professor Mohammed Racha, great guy from Kafr al-Sheikh University. And I was standing on the edge of the field. This is the field. You can see all the little labels and so on in the field. And you could see our grafted tomatoes from the edge of the field. And I just turned to Mohammed and just said, why aren't you doing this already? You know, I'm like a beginner in this tomato world. And I just said, why don't you graft? And he just said straight back, because the seeds are too expensive. And you go to the Bayer catalogue, you try to buy a few thousand seeds of their Maxifort, they're 20 or 30 cents each. 20 or 30 cents each. And you know, these guys are only getting like $1 worth of tomatoes off one plant. So there's zero chance they can invest 20 cents in a second seed, let alone go through the expense of the actual grafting process. So while I was standing there on the edge of the field in Egypt, I thought, why don't we tetraploidize these? Why don't we immortalize the hybridization and see if we could then have much, much cheaper seed production? So that was my original driver. And it's actually worked. And that's why I'm standing here very happy and still really engaged in science. We even found that this hasn't been done before, so we even patented this process. The process is incredibly rapid and incredibly spectacularly successful. So here's a second field trial, also in Egypt, a little bit further south in the Nile Delta, but still up there in northern Egypt, in lower Egypt. And we were getting very, very successful, very large increases in yield over the ungrafted controls. Now, you go, that's just an ungrafted control, it's an incomplete experiment, but it was enough for us to get very, very encouraged to go out and do a lot more. When we dug up the plants, look at these roots. Look at these roots. Here's the plants, here's the roots of the plants from the control, normal plants that they've been growing there for many decades in northern Egypt. There's the plant. And then when you dig up our plants, it's enormous difference, all right? The plants are on steroids, and it's quite remarkable, and it's legal. So what we did was we started doing experiments all over the world. And these are ones just from Kaust in Saudi Arabia. And this is a really interesting result because here are the plants sitting in a little field site in Saudi. And here's the normal plants by the end of the summer, dead. Well, no kidding. It's extremely hot in Saudi, extremely humid. We're right on the Red Sea coast. These are the plants which are grafted onto normal commercial diploid rootstocks. And then here's the plants on our rootstock. It's clearly a very, very big impact in heat tolerance. So that's something which was very encouraging as well. And we started, and yield is very, very much greater, sort of like we found in the Egyptian fields. So we went global, and we started to do this work through my company in particular in multiple countries. Yes, there's Morocco. We're actually now getting some established collaborations there, but still early days with Morocco. But Spain, Portugal, Mexico, US, Chile, also now in Argentina, and so on. The company's active now in 17 countries. And when we're talking field trials, we're talking very big scale field trials. There's 250,000 grafted seedlings here from a field trial in California. And over again, we're getting very similar results. Now note, there's 3 things to note on this slide that I'll just draw your attention to. The first is that because the plants are so much more massive when they're grafted on our rootstocks, we're talking about 2.5 times bigger plant. We plant at half the density. So here, there's the normal planting density. This one, what's this one? Central Chile, 30,000 plants per hectare they grow in Chile. We were growing as at 15,000 plants per hectare. The yield, kilograms per plant, normal yield, 3.7 kilos per plant of tomato, as 9. So 2.5 times the yield. And because you're planting at half the density, you're getting 2.5 times the yield per plant, you end up with per hectare about a 25% increase in yield. So this is very, very substantial increase in yield, which we're, as I say, very proud of. I was in a field in Chile not that long ago, a couple of months ago. I love this photograph here on the right. These are the Chilean, the normal plants, and these are like the elite commercial hybrids in a proper commercial field. And there's our plants behind. So the plants stay greener for longer. They just keep producing more and more series of trusses, more branches on the plant. And you dig them up. In fact, we took a bet. This is with one of our field officers in Chile. And then this is the normal plant. And then this is the variety he chose. And this is the variety I chose, just from standing on the edge of the field. And it was so heavy, I couldn't lift it. Miguel had to lift the plant out for me. It was such a heavy plant. And anyway. So it's working in commercial scale fields where we're looking at 3, 10, 15 hectares at the moment. Now, yield increases don't matter to a farmer. Profit increases are what matters. And I showed this to the students this morning. But basically what we're finding is that when you take into account all of the costs, the input costs, not all of them actually, but most of the input costs, you're talking about the increase in yield, which is huge. You end up with the farmers making about an extra 50% profit per hectare from their fields. And that's starting to get pretty persuasive. And this is with processing tomatoes, the lowest value tomato. But they're also the tomatoes which are grown in the poorest conditions, in the most marginal lands. Remember where we're getting our wild relatives from. So this is a very exciting result and may lead to a whole paradigm shift. And then you start going, well, hang about 30,000 plants per hectare, 15,000 plants per hectare. That's still a lot of plants per hectare. You're not getting many dollars worth of tomatoes off that hectare. And if you're going to really scale up and grow thousands of plants, then you need not just millions of tomatoes grafted, but tens of millions of tomatoes grafted. And you don't have many weeks to plant all of these tomatoes at the beginning of the season, so you have to graft them very, very quickly. There are greenhouses, there's infrastructure for growing all of this because they're already planted as transplants. Even processing tomatoes are planted as seedlings, but you need grafting machinery. Humans, fine, that works in some jurisdictions with very low salaries, but for a lot of places we need to go to robotic grafting. And by complete dumb luck, to be honest with you, in parallel with us developing our genetics in a company, Morningstar, it's the world's biggest tomato company in California, in collaboration with a Silicon Valley smart company with all of the machine learning and computer vision and so on, they've developed a machine now which can be run with one operator and will graft 4,000 plants per hour. You do your math, you don't need many of these machines to fill, in this particular example, Central Valley with grafted tomatoes. It is becoming possible. So that's my little bit of excitement where we're trying to develop, doing both domestication, if you like, with neo-domestication using wild relatives with proper commercial delivery. The final cherry on the cake, if you like, is we're not just now doing tetraploids, but we're hybridizing the tetraploids as well. So you can make one set of tetraploids, there's 2 parents, make a tetraploid from the hybrid, and then you can do the same again. 2 different parents, hybridize them, tetraploidize them, then get those 2 tetraploids and hybridize them. Why not? You end up with 4 chromosomes, 4 sets of genomes in one seed. And this gives you the opportunity to rapidly stack complex polygenic traits, such as, for example, here, salinity tolerance with Orobanche, it's now called Phalapanche, tolerance, so the parasitic plant, which is a multigenic, complex multigenic trait. It's never been bred into commercial tomatoes. We can do it now. Ralstonia resistance, same sort of story. Complex multigenic traits for sustained resistance to Ralstonia. That's bacterial wilt, massive issue. Fusarium and verticillium as well, fungal wilts, we're able to bring into a single plant genome. As long as those resistances are dominant, genetically dominant, we have a fighting chance. So that's what we're doing now. This is actually the lead breeder for Heinz in Argentina, looking at his plants, which are these ones, with our plants, which are those ones. So it's a good way to do business, I tell you, because they look at this and straight away you can see very, very big results. So here we're looking at the tetraploids. Remember, they're fantastic to start with. And then when you do the hybrid tetraploids, you're getting very big increases. Not always, actually. There's one exception here. But in almost all the cases, you're getting really good increases in the yields, further increases in yields. Okay, as well as the ability to stack traits. It's not all roses, it's not all perfect. What we do see sometimes with some of these synthetic allotetraploids is genomic instabilities. And here we're seeing some plants, this is in a greenhouse in the Netherlands, where the apical meristem, what they call, goes blind. It stops working. So I don't know how much of this is genomic instability, how much of this is an epigenetic instability. We've got one PhD student who's starting her work on this very specific issue, which we still need to address. So we're able to do very, very cheaply, and I'm talking less than $100 now for a genome resequence. You can do the whole genome resequencing, and you can do it through the generations. And so we're planning to do this for as many as, 5, 6, 7 generations. We've got the genetic material being burgeoned today. And you can see here in the F1 hybrid, you've got everything's all nice, 50-50 representation in a particular genome from each of the two parents, that if it was stable, it should stay like that through, and it's clearly not. You're getting separation of the green lines and the red lines from the middle, so we're clearly getting some non-homoeologous recombination events. We're not seeing too much aneuploidy, fortunately, but we do see non-homoeologous recombination. And that's something we need to understand the molecular genetic or genomic basis of and try to avoid in the future to get these lines stable. But otherwise, things are looking pretty interesting. So that's just an exposure to some of the research that we're doing. We're focusing very much on these tomatoes now because we're finding it could be a very powerful advance for, on lots of fronts. But these are just a couple of the take-home points that I think we could make, that I think near-domestication provides many good opportunities to generate step changes in crop production. And I think that we can accelerate near-domestication through genomics and also, of course, through tricks such as I've just been showing you today with grafting. And I think the development of rootstocks is applicable for many, many species and a lot more than we thought might be possible to also deliver some of these changes from wild relatives. These interspecific allopolyploids, I think, can generate powerful lines with very cheap production of seeds, which is going to benefit people in marginal lands a lot. And I think further hybridization of these synthetic allopolyploids can provide what I call super heterosis, because you're getting 2 sets, 2 rounds of heterotic events, as well as, of course, giving you vigor just from raw polyploidy. It also makes it more attractive to the seed companies, who we are ultimately dependent on for the delivery of seeds to farmers, because they can then protect their seeds and keep it as a trade secret what's inside their seeds, which is fair enough. And I think that this does provide opportunities for serious paradigm shifts in quite a few agricultural systems. So I'd just like to end by thanking my fantastic laboratory. It's a lot smaller than it used to be, but it's real quality. And we've got some fantastic people. We've got Natalia here. She's working on the Salicornia in particular, and now changing over to some mangrove work. And I'd like to thank them all very, very much. And I'd leave you with just a final advertisement that we are recruiting quite a lot more faculty positions if people are interested to approach me later. Thank you all very much. Thank you, sorry.
Oh, Professor, you will stay with us, please. Professor, please have a seat, please. Thank you very much. On peut l'applaudir encore une fois. Quel applause again. That was so rich. Please come back, because we're having some very important people will be joining you. Got a lot of talk to do tonight. We don't have that much time, but we will do it anyway. Ladies and gentlemen, with us today in Morocco, Laayoune, for this exceptional international Laayoune Forum of Biosaline and Arid land agriculture. I'm pleased and I'm honored to call on stage. She's section head, plant breeding and genetic section, joint FAO, EIEA Center, Nuclear Techniques International, Atomic Energy Agency. She's from Austria, and more to say about her. Ladies and gentlemen, please welcome Dr. Fatma Sasso. And we will also call, ladies and gentlemen, all the way from Utah, USA, Professor of Genetics at Birmingham Young University in Provo, please welcome Professor Rick Jelen. And of course, ladies and gentlemen, we have more seats free for the Professional Fellow at Murdoch University in Western Australia and Senior Principal Soil Scientist in DPIRD of Western Australia, Professor Edward Barrett Leonard. Wonderful. So let's start with the first question, and it goes to Mark Tester. This is your question, Mr. Tester. So, given your extensive work in genomics, could you highlight the most advanced technologies that could accelerate crop improvement? Can we have the mic ready? Is it working?
Hello,
There
yes.
we go.
I think you've heard enough from me, so I'll keep this very, very brief. I think for normal breeding programs, genomics is really most powerfully deployed using genomic selection. And genomic selection is where you're able to look across the whole genome and select for patterns. Again, we're using, this is accelerated by AI. I mean, we haven't talked about computing power and how this really is changing everything. But for young people here in the audience, I shouldn't look at you, I'll look at myself.
Look at that photo.
This is a fabulous tool for the future. You combine genomics with artificial intelligence, and breeding is going to be accelerated, and I think it's a very, very exciting prospect.
Thank you very much. Thank you so much. A question for Dr. Fatma Sasso. How do nuclear techniques enhance plant breeding compared to conventional methods?
Thank you so much. Is it working?
Yes.
Thank you so much for the question. Mutagenesis is a very powerful tool. It can create novel genetic variability to create external genetic novel germplasm for breeders. And also combining with genomic tools, AI tools, is a very strong and very important tool for plant breeding.
Okay.
And I can identify some important benefits of mutation breeding or induced genetic mutation breeding. As I said, first one is creating genetic variability. Okay. Second one is faster. When you compare to traditional breeding methods, and it is much more faster to obtain homozygous lines, also stability. Third one is, I think, we can apply for all crops, vegetatively propagated crops and seed propagated crops, all crops, especially when traditional breeding has challenges. For example, banana. Banana has chromosomal barriers, which is not possible to apply traditional breeding. And mutation breeding offers unique solution. Also for finger millet and safflower has very unique floret structure, which is not allowed to traditional breeding. And so I will say very shortly, combining induced mutation breeding and new technologies, genomic, phenomics, and also AI technologies, very strong breeding method for current, also future as well. Thank you.
Thank you so much, Doctor. A question for Dr. Rick Challen. So you have extensively studied quinoa's wild relatives. Do you believe leveraging wild species is a valuable strategy to enhance crop stress tolerance?
Is this on?
Yes.
So I appreciate the opportunity to be here and the invitation from the committee. I think in the case of quinoa, using wild species is going to be essential for the long-term viability of quinoa, at least if quinoa is going to become what I would hope would happen, and that is a staple food crop throughout the subtropics and maybe even into the lowland tropics. It's going to be essential, and the genetic variation, the natural genetic variation in quinoa is already there in its wild relatives. I'll be talking about this more tomorrow morning, but even at the tetraploid level, as Mark showed with the Chenopodium berlandieri strain that I had collected back in 2009 in growing on the shore of the Gulf of whatever it's called.
Thank you very much. Thank you. So we were in Saudi Arabia, then Austria, and then the USA. Let's go to Australia and have a question for Dr. Edward Barrett-Leonard. Australia is among the countries most affected by salinity and acidity. How has Australia addressed these challenges?
Great question. It's been a challenge, actually. Salinity in Australia is one of those things that goes in fashion and then out of fashion. Mark has experienced this. I think that we're probably in a situation at the moment where salinity is a bit out of fashion. But the truth is that many of our soils are naturally saline. And I think that we need to develop better ways, and we're not there yet. We need to develop better ways of identifying land capability and matching plants to soils. And it's something that I've really had a focus on for my entire career. It's something I'll talk about tomorrow. There are really key things that we need to look at. The challenge increasingly, not only for us, but I think worldwide, is that we need to be able to develop strategies for rapidly assessing the capability of land and targeting the right plant to the right soil.
Absolutely.
And those solutions are not immediately obvious, but I think that we are now starting to get a really good handle on it. Internationally, I think that we are heading, in some respects, for a climate change catastrophe. The world is going to start becoming much more saline, much more rapidly than we would like, and we need to have a very well-developed cohort of people who are able to provide good advice, grounded advice for farmers to achieve better outcomes on the ground. I don't think that saline agriculture fails at the moment in the world because we don't have enough great genes in plants. It fails because we are not telling the stories that farmers need to hear, and there is a real communication gap between what we know and what farmers appreciate, and we need to do much better in that space.
All right, thank you very much, Dr. Edward Barrett-Leonard. We have time for one more question, okay? Let's go with Fatma again. I got a second question for you. Could you share some success stories from mutation breeding initiatives?
Thank you so much for the question. Yeah, we have currently more than 100 countries collaborating now. So and ongoing plant mutation breeding at least 100 countries now. So this work, mutation breeding is a century actually. It is not new, and it has been applied more than, about a century. So as a joint FAO-IEA center, which I'm representing today to the IAEA, and we have more than 60, now about 60 years with establishment of our organization. And we have ongoing and also past mutation breeding programs. According to our database, 3,400 mutant varieties are being released in 75 countries to 228 species. And if you allow me, I can give a few examples from the countries. For example, Vietnam. I will give the example especially for the salinity and drought. Rice is very important for Vietnamese people. It is not only food. At the same time, their export production. So salinity, they have a very big area. But affected with Mekong Delta. The area name is Mekong Delta. Is affected by salinity. Breeders have just released a variety which has salinity tolerance. And now are being cultivated more than 1 million hectares. The breeder institution is awarded as a science award because of it increases their export. And also for soybean, it's 1.5 million people in farmers in Vietnam is cultivating mutant soybean varieties which has better resistance to drought condition and also has better nutritional quality. If I have time, I can give some examples from Africa, since we are in Africa. Recently, we are working with Namibia. This is a very new example, and their first breeding program, which prior to our program, they never had any plant breeding program. So we work together, and they are very successful actually. They released 5 copies, 4 sorghum varieties. You know that Namibia is one of the driest area in the world. Yeah. So the varieties are at least 20% higher yield than local varieties. So also one of my counterpart, Kuwait, is here. We are working with them for wheat and barley, salinity and drought tolerance. And if I have time, I can give more examples from Bangladesh or also Zambia, Zimbabwe, or coffee improvement, Sudan groundnut improvement. I have very actually good examples to share with you, and looking forward to all of you to enhance our collaboration. We are joint FAO-IEA Center, open to all member states to collaborate more, and specifically from my case is crop improvement.
Okay, Doctor, thank you very much. Do you have time to go home? No? Thank you very much. That's big numbers and a lot of countries. Thank you very much, Doctor. All right, one more question for Rick Dillon, if you don't mind. All right, could underutilized or orphan crops play a key role in achieving food security, particularly in marginal environments?
You know, a number of years ago, I read Jared Diamond's book, Guns, Germs, and Steel. And I don't know if some of you have read that book, but there's some question about Diamond's qualifications to philosophize on anthropology and plant domestication and so forth, coming from a background as an ornithologist. But that book made me think about the question, why did our ancestors domesticate the plants that they did? Why did they grab certain plants out of the wild and choose to domesticate those plants? And should we be beholden to their decisions that they made thousands of years ago? And I believe that as scientists, that's a fair question. And I'm one of those who have been involved in neo-domestication of crops, not only quinoa as an orphan crop, but also a species of oat that's native to northern Morocco, Avena magna. And I am convinced 100% that nature has many, many lurking secrets, salicornias and other plants that could be tremendously beneficial to humanity, especially as we move forward into this uncharted territory with climate change.
Thank you very much. And the last question goes to Professor Edward Bart Leonard. Given that soil salinity, sedicity is a critical issue in Morocco and elsewhere, what are the most effective on-farm practices to mitigate its impact and sustain productivity?
Such a good question. Sedicity is caused by, it causes soil dispersion, which means that water doesn't infiltrate into sodic soils very well. One of the consequences of that is that with a condition of waterlogging if we apply water to soils. And waterlogging in saline soils is a very nasty combination because it causes oxygen deficiency in the soil. Oxygen deficiency causes an increase in salt uptake by plants, and plants will die. Now, a traditional way of trying to fix sodic soils is to apply gypsum. And we have just in the last couple of weeks published a paper based on sodic soils in Australia, which are very alkaline, that suggests that one of the reasons that gypsum may not work in these soils is because they naturally have high concentrations of sulfate, which prevents the dissolving of gypsum, and these soils also, because of their alkalinity, have high concentrations of carbonate and bicarbonate, which gobble up the free calcium and precipitate it out as calcium carbonate, which means that the soil, the gypsum doesn't have the beneficial effect. Okay. So in addition to trialling gypsum then, I would be looking at trying to address issues to do with ameliorating waterlogging, and I would be, and one of those is the operation of very shallow drainage. We're not talking about drainage that is sufficient to lead to the deep leaching of salt, but shallow drainage so that the water doesn't lie on the soil surface where plants may be located, but runs into an adjacent furrow. The water sits there, it's available to the plants, but doesn't cause waterlogging. A third thing that I would be trialling is the use of mulches, and I think that mulches more broadly, and we have a huge amount of straw worldwide which is burnt at the end of cropping cycles. Yep. Why not keep it? Why not lay it on the soil surface where it decreases capillarity and can keep soils moist? So a range of management options apart from gypsum, which is the traditional method of ameliorating soils, may help with the treatment of sedicity.
Well, thank you very much. Professor, Doctor, thank you so much for sharing your knowledge, for these exceptional answers. I think it's time for a cup of tea and a coffee. All right, and we'll come back. Thank you very much, ladies and gentlemen. Let's go for our coffee break, and I'll see you at 5 p.m. Thank you very much. Ladies and gentlemen, please be seated. We're starting in a few minutes. Thank you very much. Merci de vous asseoir. Nous commençons dans quelques instants. Merci. Well, ladies and gentlemen, I hope you enjoy your coffee and tea. It's, again, an honor and pleasure to have you here in Morocco, Laayoune, for this exceptional event, the third International Laayoune Forum on Biosaline and Arid land agriculture. Ladies and gentlemen, we're starting the second plenary session. From lab to farmer, how to fill the gap and accelerate the adoption. And with us here today, starting with the first guest that we have, Professor Rachid Sarraj from UMCSP Morocco. We will also call Professor Mina Devkoda from ICARDA Morocco. You have a keynote, yes, sir. And we also will ask Mr. Abdurrahman Amri, Regional Department of Agriculture, Laayoune, Morocco, to join us. Be seated. Mr. Bakad Mohamed Lamin, Halib Saqi Al-Hamra Cooperative, Morocco. And Mr. Naufal Rudi Al-Mutmir, UMCSP, Morocco. And of course, let's start with a keynote speech from Professor Rachid Sarraj.
Thank you, sir, and thank you, colleagues from Asari. This is the second time I came to LAFOBA. I was here 3 years ago, and I am very glad to see the progress, not just in the quantity of people, but also in the themes. Now, I want to shift gears compared to the first session. I like very much the title of How Do We Link? Because science has to have a purpose, at least in our context. So we are in Africa, and obviously, science is in the middle of our challenges. So what I try to do, a little bit touching on my personal experience, as much as possible, case studies, because we don't learn, from my little experience, I don't think we learn only from concepts, but we learn from experience. I have been around from different continents, different contexts. I will just give some quick snapshots. How do I move this thing? There you go. So this is what I'm going to be talking about today, my main messages or outlines. First of all, I think I'm kind of inventing a new term, this R4D for I. Just an attempt. What is it? Is research for development. That's common, R4D. Now, R4D, if we want to take one step further, we take it to impact. And I'm not going to give you any theories about it. I'm just going to give you some examples, 2 examples. One on abiotic stress in rice, and you know the importance of rice for food security in Asia and now also in Africa and other parts of the world. And the second example, I'll be talking a little bit about salinity tolerance and germplasm at ICBA. And then I'll go into presenting some sort of a framework of a systems approach, because we're dealing with something that's really complex. We cannot dissociate it. We have to look at the integration of the system. We dig a little bit into the topography of crop yields, because this is what it's about productivity. We need to understand how do we work on that, especially in the context of Africa, again, because we're still very far from the potential. And first topic is very close to my heart. In Arabic, we say the first love is the one that you keep for all life. And for me, symbiotic nitrogen fixation was my first love. And it's still very, very close to my heart, especially because I see it hasn't yet reached its full potential. So again, very quickly, I'll touch on that because I believe is a huge potential for that in Africa. And finally, I'll tell you just in a snapshot what we're trying to do about this at University Mohammed VI Polytechnic, based on our strategy. So let's move on. Oops, there's something wrong that happened here, yeah. The slide was supposed to come just once. That's the sentence in red. This is the little story. We were all working at the International Rice Research Institute in Los Baños, IRI. IRI is one of the centers of the CGIAR who made the Green Revolution. These guys have, since the 60s, 1960 to be accurate, started working, developing research. And we had teams, unfortunately, Dr. Abdel Ismail, who was supposed to be also here to talk about salinity, couldn't make it, but I'm gonna cover a little bit for him. We had the team working on drought that I was leading, and you can see a book there on the right. If you're interested, it's available online, free. It's called The Rice and Drought Frontiers, because we were trying to do the science of how do we break these barriers of yield in rain-fed environment and drought. And then one day, we got a visitor from the Gates Foundation. Historically, now I'm talking about 2006, 2007. Gates Foundation, you know, they started in health. They knew nothing about agriculture. Then they started coming to agriculture. They funded one project on maize, drought tolerance in Africa. And then they came to rice in Asia. So we had this guy who ended up being one of my good friends, Prabhu Pingali. Some of you might know him. So Prabhu listens to us the whole day. We give him PowerPoints, and he say, okay, good. And at the end, he says, if we bring some money from the Gates Foundation, how many farmers do you think you can reach? That was his question. And to be honest, none of us was prepared to respond to that question. So we said, well, can you give us a little time? In the evening, we went with the economists, people working on salinity, and tolerance, and submersions, and we started, and we said, oh, we think we can reach 10 million farmers in 10 years. Because we're talking about rice in Asia, and you know the importance of India, Bangladesh, Nepal, China, you name it. Prabhu is a very wise man. He listens to us, he says, yeah, that's good. How about in 3 years? That was a tough question. This guy is talking about reaching farmers in 3 years. You have to get something ready already to move. We went back to our calculations and economists, and following morning we said, we think we can reach 350,000 farmers. And it has to be documented. He said, great, go ahead and write the proposal. It was for $20 million. Now, what we sold to Prabhu and the Gates Foundation is a proposal to improve salinity tolerance, drought tolerance, and submergence tolerance in rice. In 3 main countries, in India, Bangladesh, and Nepal. So we started that phase. This is what this story is about. The project is called STRASA, Stress Tolerant Rice. And I'm showing this because to me, my little humble experience, this was really a learning lesson. You can have impact. It is possible, especially if you get the pressure from your donor, and the donor, like Prabhu, knows exactly what to expect. Because it came in the context of 2008. So you may remember in 2008 what happened, the food crisis, the economic crisis. The government started pushing, like in India and Nepal and Bangladesh. They were basically waiting for germplasm, for technology, so they were pushing. As soon as we were putting something in the pipeline, it was moving in the pipeline. And guess what? After that first phase in 3 years, we've exceeded that 350,000. It was way above that. So Gates Foundation was happy. He said, go ahead for a second phase. And actually, that STRASA project ended up 12 years. until 2019. If you can look at the website, you can see the numbers, and it's all documented. Millions of farmers that have benefited from these technologies. I was going to show the video, but I don't think for the sake of time, I'm going to escape it because it shows a little bit the elements. So salinity tolerance, drought tolerance, submergence with the sub-1 gene, and the impact was there, and I think to me is one of the best cases. So now the work has to start looking at the targets. And this is a key message I want to send today. If you want to have impact and linking what we say to farmers, you have to have very clear idea of your targets. Where are you going? So what kind of target population of environments? You characterize that environments, you know the soil, you know the climate. If you don't, you're going to be most probably just shooting in the dark. And I'm saying this because I think today would be about the right time to do a similar example for Africa. Africa has not had something similar for different reasons. So the drought-prone environments, just the drought-prone is 20 million. If you add the salinity and the submergence, it adds more. They have what they call these mega varieties, like Swana, IS64. Mega varieties are varieties that have been bred at Erie, but that have expanded very widely across Asia. But the problem is they were stress sensitive. So now the question was that, should we do the breeding starting from scratch and the agronomy, all that, or should we build on that and try to introduce? And the big idea at that time, if you remember early 2000s, was all the work on molecular biology, bringing QTLs, bringing candidate genes and all that. For instance, on submergence, we had the sub-1 gene. So it was relatively easy to insert the sub-1 gene in the Swana, and it spread like fire in the bush, ready to go. So kind of cutting in the timeline that it would take to the pipeline. And this is the approach that was used in all this work. The advantage, so that supposes also knowing very well the environment, as I said. So in here, and I think if some of you are using this, and I saw some talks will be talking about this characterization through satellite. This is 2007, 2008. We use electromagnetic resonance to map the soils because you have lots of variability. So if you try to superimpose genetic variability on soil variability, you get lost. There's absolutely no way to get from this in the field. So you need to have kind of a covariable with the environment. And this is the idea. There are several ways. Now, the slide was supposed to come one at a time, but it didn't happen for some reason. We don't bother. The starting point is that slide on the right side. In Jharkhand, India, we were working mostly in Eastern India because Eastern India are drought-prone, mostly rain-fed, yeah? And it's one of the poorest of the poor. You have people who are today farming, tomorrow they might be doing something else, selling shark oil or something, because they are in the edge of poverty. So here the work on drought is not just improving crop resilience, but it's improving livelihoods of people. That is the starting point. And this is somehow how I try to respond to that big questions, how we link research to farmers. What I say, it has to be starting at the farm. Know the farm, know your target, know the client, know his problems. And from there you can go in backwards. Working with the NARS for trials, station, that's what you could see, the Erie field, where you can develop all the technologies. And moving up to, for instance, at that time, we had a very strong collaboration with François Tardieu in Montpellier, where he had all this phenotyping, very accurate. But what I'm trying to say, don't do it. Don't try to do it the other way, because it doesn't work. A lot of researchers have this dream, I'm going to develop this thing, whatever technology in the lab, and then we're going to move it. It doesn't work like that. Most of the time, you have to put farmer in the center, and start with him if we want to make impact. That's the message. Very difficult interaction with the environment. So the end result, this is just an example. We developed this network. This was basically with Gary Atlin, who is now at the Gates Foundation in Seattle. You might know him if you send him proposals on crop breeding. He's going to be most probably the guy who's going to review it. So Gary has, and as a team, we work together with the NAR. So all those little round green circles, those are stations across India, Thailand, Nepal, and Bangladesh. So how does it work? Very simple model. We have this shuttle breeding, as it's called. You move material back and forth. So you send it to the stations, we collect the data, we do multiplication trials, we collect it back, and it goes back and forth. And the strength of a network is that once you develop something interesting for that particular environment, it moves directly to farmers' fields. So, and we had varieties, and you see examples there, RB6, Shabagidan, and so many others. So this model has worked. It's called participatory. Sometimes in the jargon we call it participatory drought breeding network. It can be participatory varietal selection, but I insist a lot on participatory. Because here what's meaning is the farmer is part actually of the team that does the work. Now, I shift gears, move into salinity. As Mark was putting it together, this is, you know, with time flies and we grow older very quickly. So this goes back to the initial days of ICBA. ICBA started something around 2000, 1999, something like that. To be honest, they didn't have much to start with. They were working on halophytes and things like that. At that time, I was the privileged, I was posted in Hyderabad and the headquarters of ICARDA, in charge, basically, of screening material for drought. So we looked at pearl millet, sorghum, peanut, chickpea. So we got this visit, interaction with ICBA and said, hey, you get some material. We started screening for salt. We published a couple of papers that are up there, both on sorghum and millet. The particularity here, the nice thing that was developed at ICRISAT was that they have what they call a mini core collection. So what is a mini core collection? It's somehow about a panel of 200, 250 varieties that represents the whole range of variability you have in the crop. Because you cannot bring, I don't know, 60 or 70,000 entries. It's impossible. But if you could bring that into 200 or 250, it becomes doable. And it was the time where molecular markers were coming in a big way. So the big dream was that we're going to do didn't work out all the time, isn't it, Mark? But the bottom line is we were able to identify salt-tolerant material, both on sorghum and millet, and it has moved. I didn't know at all what happened to it afterwards until I visited ICBA much later, like 10 years later, and I talked to the people. Ismahan El-Wafi was the DG, and he said, yeah, we have impact and things, and I start looking at that material, and I say, come on, this came from my lab. Yeah, so, because what they have done to it, they have moved into several countries, Egypt, Jordan, Oman, Syria, Tunisia, and they had the second phase. But originally, that material started from the screening we did in Patancheru, India. A very nice story. What I'm trying to make out of this is, again, for impact, it has to start from the right targeting of problems and look at the variation. Some would say, the agronomists would say, yeah, breeding is the easy way because you just move in varieties. If you try to do this in agronomy, it becomes much harder. But the approach we had both at ICRISAT or at ICARDA or at many centers is we try to integrate as much as possible the breeding with the agronomy, with the socioeconomics. You have to have everybody on top. If you don't have a team, multidisciplinary, you're not very likely to make impact. That's another message. Now, taking a broad picture, so this was co-authored by my friend Prabhu Pingali, who was our donor to the Gates Foundation. In here, it's a completely different setting. We're trying to look at the parameters. This is a forward-looking exercise that we did at the Science Council of the CGIAR, looking at the problems. No big surprise. You know, I'm sure, all of them. Yeah? You know the pressures, and you know also the ways we're going to come out of it. The culprits are demography. We hear all the time, especially if you put it in the context of Africa. Our demography, trade, energy. If you take just trade these days, for instance, it's becoming a very, very important research topic, isn't it? With all the disruptions that are going to be happening in international trade, and how is that reflecting on food security? The one that we spent most of the time, I did spend most of the time working on, is climate change. So here is, in a nutshell, my story with the climate change. And unfortunately, it didn't come with the slide because it was supposed to enroll. In the background of this slide, you will see a couple of papers published. One of them published in one of the previous numbers of global change biology. And we started looking at how CO2 increase and temperature interact with drought and other factors on productivity. At that time, I was working with the US Department of Agriculture in the US on soybean. So CO2 was around, the ambient CO2 in that time, I'm talking about mid-90s, 95, 96, was about 360 ppm. I checked it before coming here. In April, it's 429. So we were comparing normal CO2 to high CO2. The point I'm trying to make here, when we started doing all this work, the COP mechanism, the Conference of Parties on Climate Change, was not yet done. It started in Kyoto in 95, and then it's Berlin, Geneva, and it started moving down. And you know that this year we will be having COP30 in Belém in Brazil. What is the lesson? The lesson is that, again, it's another entry point to impact. If you don't link science and policy, you're most likely to be wasting time. And this has been a dilemma for me for over years. Science is, of course, was preceding, because in the 90s when we started doing all those phase experiments, there were still a lot of people who have doubts, either if climate change is happening, including in the scientific community in the US, that people say, well, doubting about it. Now, what I'm trying to say is there is a huge gap between the science and policy, and if we're going to do properly our job as scientists, I think we have to do as much as possible to link the science and policy dialogue. And what is better than climate change as a topic? This is an invitation to make your points for the COP30, hopefully, because this is becoming a crucial time for humanity as a whole, and especially the responsibility of the scientists to show where do we stand. And if we put it in the context of Africa, we stand on a big call for adaptation. Because without adapting agriculture, African agriculture to climate change, we're all dead fish. Sorry for the expression, but that's how it goes. Adaptation. Now, I go back to this model. Some people spend all their lives looking at complex models. I particularly love simple models. Simple, mechanistic. This is a model from Tim Sinclair, with whom I used to work in the 90s. It just shows productivity at crop yield like you're trying to escalate a mountain. So we're down here, you try to go to the top. You cannot jump to the top. You need to go scale by scale. So the equation in the middle is just how yield is made. Very simple. It's basically just how much biomass you get with interception radiation, carbon, and harvest index, which basically partitions part of the biomass into seeds. Now, what are the constraints? Some people will tell you a zillion parameters, but actually, if you want to make a good yield, farmers know that already. Control water, control nitrogen, and phosphorus. You've done it. Most of it. Of course, you might still have micronutrients. You still, I know my colleague, Mustafa, will be mad at me because he's pathologist. You look at insects, you look at disease, you look at, but the bottom line of productivity of the topography of yield are these factors: water, nitrogen, and phosphorus. So let's start with the water. Again, this was a slide that's supposed to be rolling. It doesn't. Africa is, as you can see, the map. This is data from FAO and UMI. Very, very low still today, potential irrigation. Yet we know that productivity, if we're talking about drylands or soil environments, whatever, the bottom line and the bottleneck is water. What does that mean? You could look at the tables on right side from IMI and you see how much increase you could have in yield just by bringing water. Problem is availability of water. Where does it come from? Sometimes in other places, there is no other way. In groundwater, you could look at rainwater harvesting. We look at shallow groundwater. I'm sure this will be discussed in this conference. But to me now, after all these years, what I'm saying is that science has made a lot of progress in some areas. If we take, for instance, water harvesting, ICARDA has spent years and years in developing the technology. My friend Dibawis has spent all future, because that's how we're going to unlock the potential of productivity in Africa. How do we bring this all together? Is integrated soil fertility management. This is not new, again. Technology has been developed. Most probably, I mean, during the old times of TSBF in Kenya, ITA in Nigeria, and many others. I think the technology, again, is now how to bring mineral fertilizer with organic, with farmyard manure, and all these technologies. You see that it has been developed even more than a decade ago, but it's time to roll it over in Africa in a big scale. This is, again, a theme for, and it involves, of course, biological nitrogen fixation and farmers. I'll go back to it. This is what I mentioned my first law. The reason is that nitrogen is the one that has made possible all this increase in productivity in the 28th century, and maybe even in the demographic increase, because since we discovered the Haber and Bosch technology to synthesize the nitrogen, things become possible. But you have it, you can see the numbers. Biological nitrogen fixation is about 200 million tons per year, and it's free, comes with the nature. We have not yet exploited all the potential. I've got to go fast because I see Abdelaziz looking at me. Related to that gap, legumes, you see the green revolution since the 60s, that's in red. You see what happened in the productivity. It has increased. This did not happen with other crops like pulses, because the nitrogen fixation happens mainly with the legumes. We can, again, have a whole workshop, I think it was in 2016 in Marrakesh, the Pulses Congress, discussing this. Why we haven't been able to move this green curve to become similar to the red one? Several reasons. And we have also ignored the wide range of variability. Those guys with the star, I discovered them, for instance, when I was doing my trials on pearl millet in Rajasthan, in northern India. My job was not to work on mothbean or all those, was to do the trials on pearl millet. But you see these pulses on the edge of the desert of Rajasthan who are able to produce and to fix the nitrogen from the air. Isn't that wonderful? So this is an invitation to our friends in Asari to consider some of this work in the future. I think, and I believe, it holds very large potential. We can go in discussion. This is a slide that I presented a long time ago in that workshop on pulses. Why we haven't resolved? This part of the response is that it's not only technology. You have policy. You have, legumes have not received subsidies, for instance. They have not received any push from them. And maybe it's time to reconsider all the life cycle of all this, because it's proteins, it's nitrogen, and it's also most probably ecosystem services, but the work is still to be done. I'm going to zip this quickly, work that was done on nitrogen fixation, because the bad news with nitrogen fixation is that it's extremely sensitive to stress, both salinity and drought. And that's why I'm showing that here. And I hope that some focus of the research has to go there. We've done some, and you can see there in the trials that were done in the field, where you see selections of soybean resistant to drought with higher yield and also with higher nitrogen fixation. I think this needs a little further digging in our context and in the African. This is more than 20 years ago. The proposal was there to have a mega program on increasing in the tropics across Asia, Africa. So we had all the CGIAR centers, NARS, and unfortunately the CG was not ready for funding, but it happened into Africa. Again, I invite you, some of you to look at this, look at the results. This is Ken Giller, Professor Ken Giller from Wageningen University, who has done an amazing job all across Africa, bringing into fixation in the field. You can see some of the data on the impact it had. And the most important, as Mark was saying, is when you put it into dollars. So this was bringing improvements in livelihood. I'm cutting a long story short, I'm sorry. I'm not giving you all the science behind it, but a lot of it is not even rocket science, as we say. A lot of it was inoculation. In cases, for instance, where the rhizobium was not in the soil, you bring it up. You bring the P when it's needed to enhance the symbiosis, and you increase the productivity. To cut a long story short, the impact was evident. The project now has been also by the Gates Foundation for 3 or 4 times, but now it's over. It needs maybe a new insight and a new team to take it forward. I'm just pointing to possible directions in the future for Africa. This would be one of them, because it has already proven success, and the need is out there. And we do work on it at UM6B. Now, the slide did not roll out, so you don't see all the slide of the strategy of UM6P. This was a collective work that we've done at the University Mohammed VI Polytechnic. It took us about a year and so, including all the team working on soil, working on plants, working on crops, working on water. And we came up with an integrated vision and strategy that links the research. Unfortunately, we don't see all the details, but you can check it. It's available online. That connects the research down in the labs to a higher level of impact that could go even to the SDGs. And in the middle, of course, we have to work out all the interactions. One of those strategic pillars is SP4, and see Harish was very instrumental in developing the concept, is on dryland agriculture and marginal environment. And I would like to highlight again those strategic priorities. Number one is crop livestock production systems. So if you do, and I think ICARDA does a great work. I used to be the director at ICARDA of diversification and sustainable intensification, and we did have a livestock program that's really important there. It's a must. So because that's how people live in the drylands. And generally, they don't do just monocultures or cropping. They do crop livestock. So we have to bring that integration. Agroforestry, this is becoming fashionable today for different reasons. Carbon markets, why not? If it can add a little more incentives for the farmers to go in there. But I think it's really important to start bringing the science of agroforestry in there. And finally, agronomy. Agronomy does not get old. That's my take on it. Technologies evolve, but agronomy, the basics of agronomy still remain the same, and they still have the promise of making a big difference in Africa. Just the basics of managing water, managing nutrients, and bringing the socioeconomics, because sometimes we forgot this side. And an idea that came in that concept is an observatory. So maybe it should see the days, the light at Asari or with some of the work. And with that, I leave you with my take-home messages. I'm sorry, I've been a bit quick, but these are my take-home messages. The first one is, I hope I did convince you that R4D for impact is possible, but it needs to be systemic. You have to look at, you can't just take a plant in a lab and hope for impact. It has to be systemic. It has to be participatory. If you want farmers, you need to include them from the beginning and know exactly what's their problem. It has to be targeted, and it has to be planned. You plan it ahead of what kind of improvements. Touching on climate change, I mentioned the important role of science policy dialogue. I think we still, at least at UM6P, we still kind of learning how to get there, because partly our policy center is in Rabat, the technology is either here or in Ben Gurion. I think we've got to put our act together and see how do we bring it, how do we integrate science and policy. I hope I've convinced you that sometimes, especially dealing with water, technology is there, and water harvesting, supplementary irrigation. What it needs a team to bring it to scale, and a commitment to bring it to scale in the drylands. I wanted to, and my dream is to see that in large areas in Rahamna, but also probably here and other places. Because you get this year, for instance, when you look at the rainfall, you can have many, many months of drought, not a single drop, and then suddenly, in one day, you could receive 100 millimeters. It happened even in places like Rashidia this year. And all that water gets lost. If we know how to manage this, and I think the technology is out there, it's very important and crucial. On nutrition, of course, we talked about the integrated soil fertility management. It has been demonstrated, it has been proven successful, but it is also context specific. It's not a magic stick. It has to be adjusted to the local conditions. And finally, I try to bring the point about the UM6P strategy. We have a very ambitious vision, not only for Morocco, but for the whole of Africa. We don't see ourselves as lonely players, but we work with others, many others. We work, for instance, with ARUA, which is the Alliance of Research Universities in Africa. We work with and I will finish basically by inviting you this summer to the African Association of Universities. This association was created in Rabat in 1967, and now we're bringing them back. There will be about 450 universities from 20 July to 25th, and I hope many of you will show up there and participate, because Africa is now trying to put its act together. This is happening. And UM6P trying to be one of those actors to push in that direction. And with that, thank you very much.
Well, Dr. Siraj, it's always a pleasure listening to you and watching the way you're explaining things and your advices. And I wish your dream come true. All right? We can applaud Dr. Siraj, please. Les applaudissements pour Dr. Siraj. Merci beaucoup. Now, ladies and gentlemen, let's start our discussion panel. And first of all, we'll keep the first question for Dr. Rashid Siraj. Seeing that you have a very long experience working with farmers in marginal environments, do you believe that in addition to scientific research, professors and researchers should make greater effort to engage directly with farmers, or is this primarily the role of extension services? You have a mic? Here we go.
Yeah, it's a very important question. Is it a role for scientists? I say, for a scientist who chooses, who decides to be working in this area of ag research for development, I would say yes. He has to be.
I think we don't hear you that well. If we can just, here we go.
Is it coming?
Yes, now it's coming.
Can you hear me now?
Here we go.
I was saying, for a scientist who chooses to be an agronomist or working for ag research for development, I would say yes. He has to be at least aware of the conditions and requirements of the farming communities. But of course, not everybody is an extension agent. When we used, for instance, to travel to Eastern India, we would go as a team. So there is the breeder, there is the agronomist, there is the socioeconomist, and we work with the extension agents. But at least it helps you, before going to the lab, it helps you coming closer to the reality of the farming community and knowing what are the bottlenecks. I wouldn't say you have to replace the extension agents, otherwise I will make my friend Naufal not fair. Yeah, everybody has to be aware of those conditions. The extension is the one who keeps continuously in touch with the community to make that transformation.
Well, thank you so much, Doctor.
Yeah.
Thank you so much, very much. So we have like 17 minutes to go. We'll try to get all of you having your questions. Let's move to Dr. Mina Devkota, Senior Science Agronomist at the International Center of Agriculture Research for Dry Areas, ICARDA, Morocco. Nice to have you here, and I have a question for you. So ICARDA conducts significant applied research in dry areas. Based on your experience, what is the most effective way to encourage farmers to adopt the solutions developed?
Yeah, thank you. Thank you very much. It's very, very interesting question. And Dr. Siraj has already covered many things, so I think I have very less to say on that line. So working in a research for development organization, almost more than 15 years, and always working with the farmers. My experience is definitely from the designing phase to the, to the scaling phase, we should to be keep farmer at the center, core center. So we should think of farmer as a core center. And for example, rather than working for them, we should to work with them. So from the very beginning, as an agronomist, especially as I said, Dr. Rashid said, yeah, there are different technologies, but most of the agronomic practices we are targeting, we should to work with them. Yeah. not for them. So this is the one most important thing I would like to highlight. And I will just share one very good example this year. I have been trying to diversify the system when I joined ICARDA here. And one of the innovation that I have been testing, like, how we can really intercrop to harvest two crops. So this year I had a very good lesson learned from doing the on-farm demonstration together with the extension and local research and with the farmer. One farmer, he taught me that, Meena, yes, you taught us to grow two crops. Now I will teach you I can add even another third crops instead of this one, because I have this limited water. For example, I was trying to push lentil and really intercropping with onion or something, chickpea. And he said, okay, now I'm harvesting lentil, I will plant melon. So what I'm saying is, once they see the advantage, once they see all this kind of thing, definitely they will even encourage for adoption, and we don't need to too much push on that time. Yeah, this is something I have very take-home message is work with them, not for them.
Dr. Meena, thank you very much. سيد المدير الجهوي للفلاحة في جهة العيون، مرحبا بك.
مرحبا بك، شكرا.
اهلا وسهلا. هل ممكن ان تعطينا نبذه عن المشاريع الزراعيه المنجزه او قيد الانجاز بالجهه، وهل تعتقدون انه من الممكن ان تصبح الصحراء المغربيه يوما ما سله للغذاء بالمغرب؟
شكرا السيد الوالي، الساده الحضور كلهم باسمه وصفاته. اولا اود ان اشكركم على الدعوه لهذا المؤتمر اللي هو عنده واحد طابع خاص، خاصه انه يعالج بعض احد التحديات اللي كنعانيو منه من خلال مشاريع فلاحيه اللي كنجزوها على مستوى جهه العيون الساقيه الحمراء. كيتعلق الامر بلا سالينيتي، شده الملوحه اللي كتعاني معها الفلاحه. ولكن بالاضافه الى هذا، فقط ليعلم الجميع ان جهه العيون الساقيه الحمراء عندنا تحديات اخرى اللي هي كبرى، تحديات مناخيه، قله التساقطات المطريه، قله الموارد المائيه سواء منها السطحيه او الفرشه المائيه كذلك السطحيه. كنعتمدوا بالخصوص على فرشه مائيه عميقه كتمتاز اولا بعمق اللي كيتراوح ما بين 300 متر حتى 1400 الى 1500 متر، شده الملوحه من 2 غرام مع صابي بطبيعه الحال اللي هو قليل الى 7 غرام حتى 10 غرام. كذلك هناك ارتفاع في الصبيع في بعض المناطق اللي كيوصل حتى 70 لتر في الثانيه. هذا كيخلينا اننا نعتمدوا خصوصا على هذه الفرشه المائيه باش نقدروا يعني نخلقوا بعض المشاريع الفلاحيه من خلال اليوم. الحمد لله وصلنا اننا نستغلوا بعض الضيعات الفلاحيه خاصه مع تعاونيات حليب الساقيه الحمراء على مستوى المدار السقوي ديال ديتيوس. خدينا بعين الاعتبار جميع التحديات، يعني الترمل، الرياح قويه جدا، ملوحه، لان اليوم كاين يعني مجموعه من الابحاث واللي عملنا على التطوير ديال هذا البحث على مستوى جهه العيون الساقيه الحمراء مع بعض المزارعين، واللي عطات نتائج مهمه في بعض الاماكن اللي ولات فيهم الفلاحه مستحيله، خاصه في الواد اللي وصلت عندنا شده الملوحه ل 10 غرام في بعض الضيعات الفلاحيه. كذلك على مستوى مشروع اللي هو ملكي بالجريفيه على مساحات كذلك 250 هكتار في الشطر الاول ديالو. بطبيعه الحال هناك تحديات اخرى بالاضافه الى الملوحه، فعندنا التحدي ديال الكبريت اللي كاين، لو سوفر اللي كاين في الماء، الحديد كذلك. ولكن كاين يعني حلول اللي جبناها من خلال التحليه ديال المياه، الاجاج. وعندنا في هذيك المنطقه بالضبط واحد منصه اللي نبغي نشكر السيد الوالي وكذلك ازاري، منصه للابحاث على الزراعات اللي يعني كتحمل الملوحه وكتستجب لهذه الظروف المناخيه، اللي اليوم اوبيراسيونيل خدامه وفيها انتاجيه مهمه، وغتكون منصه ما بين البحث العلمي الى بطبيعه الحال الفلاح والتطوير ديال هذا البحث العلمي على مستوى الميدان.
شكرا جزيلا على الجواب، شكرا جزيلا. معنا اليوم السيد بقاد محمد اللمين، كوبيراتيف حليب الساقية الحمراء. أهلا وسهلا. منطقة فم الواد كما هو معروف كانت منطقة فلاحية بامتياز في إنتاج الخضروات والأعلاف. ما السبب لتقلص هذه المنتوجات المزروعة في المنطقة؟ وما هي الحلول التي لجأت إليها تعاوناتكم لتوفير الأعلاف؟
شكرا على الاستضافة. احنا اليوم نفتخروا بهذا الحضور ديالنا وسط هاد الباحثين وهاد العلماء وفي هاد المؤسسات.
غادي غير نصاوبوا الميكرو ديالك شويه باش نسمعوك جميع عافاك. بارك الله فيك.
كنشكركم على الضيافة، وحنا الحمد لله اليوم قاعدين وسط هاد الباحثين وهاد العلماء اللي كان عندنا واحد المشكل اللي هو في مولواد في الضيعات. كان عندنا واحد المشكل ديال الملوحة. عندنا الملوحة دائما طالعة، وحنا كننتجوا الحليب وكسابة ديال الأبقار في جماعة فم الواد. إذا كان عندنا هذا المشكل مطروح من 2017. دخلت كانت واحد الاتفاقية دارت تعاون حليب الساقة الحمراء مع فونداسيون فوسبوكراع مع البحث العلمي ديال الأبحاث. المهم من خلال هذيك السنوات عندنا دريك الفلاحه اللي كنا كنديروا كنعرفوا مات لينا نقدروا نديروها لان الملوحه طالعه. اذا كان هذا التدخل ديالها الشركاء مع التعاونيه وجبروا عندنا مشاكل في الملوحه ديال الماء والمشاكل في الملوحه ديال الارض. اذا هاي النباتات اللي كنا كنديروا قالوا لنا راه ما تخدمها لكم حسب المعطيات اللي عندنا، خاصكم تشوفوا واحد الزراعه بديله اللي ديروها باش تقدوا تستمروا في هذا القطاع. اذا احنا كسابه ديال البقر وعندنا الحليب، البقر كينتج الحليب، اذا خاصنا شي نوع خدام صالح للابقار، الاكل ديال الابقار. اذا تقريبا النتائج ديال هذوك الابحاث اللي كانت دايره فونداسيون فوسبوكراع وليكبا، جات مؤسسه محمد السادس متعدد التخصصات وتبعت المراحل النهائيه ديال هذه النبته. اذا هذه النبته كيقولوا البليبانيكوم هي اللي كتنتجوا دابا في مولواد. اذا جبروا لنا على انها خدامه لنا بالنسبه لهيك الملوحه اللي عندنا وخدامه لتغذيه الابقار. اذا هذه النبته دريك تبعوها جبروها على انها صالحه بالنسبه للتغذيه ديال الابقار. تقريبا فيها 17 ديال البروتين حسب الابحاث اللي كاينه، وكنخدموها بالمكننه. اذا نحصدوها نزرعوها وكنحصدوها، ولكن الزريعه دائما خاصها تكون من الضيعه. اذا الا جبناها من بلد ثاني والله شي ما كتنجح. اذا هذيك اللي عندنا في الضيعه هي اللي خدامين فيها. بدينا فيها تقريبا شي هكتار، ودرك الحمد لله عاد عندنا 9 الهكتارات، ولينا كنحصدوها بالمكننه ولينا كندخلوها للتعاونيه حليب الساقه الحمراء. اذا هذا واحد الدخل جديد عاد الفلاح بالنسبه كيبيع ذاك الشيء اللي عنده يبيعوا للتعاونيه، والتعاونيه عندها واحد الميلونجور كتصاوب واحد التغذيه خاصه بالنسبه للابقار، ابقار الحليب. اذا في البروتين جبرناها 17 بروتين. اذا هذه يدرك الفلاحه بالنسبه لهي النبته عادت واحد القيمه مضافه بالنسبه لنا احنا نتيجه لهذه الابحاث ديال هذه الجامعه اللي عادت هون حدانا الحمد لله، وهذو الباحثين اللي عادوا كيجونا على استمرار بالنسبه للضيعات في مولواد. اذا والنتائج ديال العلميه ديال هذا الابحاث اللي كيديروا هذو الباحثين والله هو العلماء والله هذو الطلبه عندنا احنا الاوائل اللي كنستافدوا منها. اذا هذا واحد الانجاز الحمد لله اللي نشكر عليه هاد العاملين على هذه المؤسسه. وحنا موجودين، احنا راه الاشارات تجارب الحمد لله الارض كاينه بالنسبه لفمولواد وحنا جيران. اذا شي كامل بغاو يعدلوه هما فايدته على الجهه وعلى المنطقه، احنا موجودين
الحمد لله. الحل توجد وصافي الامور تقدت
الحمد لله.
الحمد لله. شكرا بزاف على التدخل ديالك. So we moving for the last question and it will be for Mr. Nofel Rodis Al-Mutmir, UNCSP Morocco. So the Al-Mutmir initiative launched by OCP Group promotes best practices in cropping, including fertilization and conservation agriculture. Could you share some key performance indicators such as the number of participating farmers, total area covered, and percentage productivity improvement achieved?
Thank you. Thank you for inviting us to this forum. Just if you allow me to say some words about
Of
Al-Mutmir. Al-Mutmir, as you said, was created by OCP and he joined UM6P in 2022. And the mission is an integrated farming development model who is providing a customized offer for our farmers. And while we have this feedback loop between the Al-Mutmir and science and the farmer. Al-Mutmir is a team, really thanks to our team. We are more than 120 engineers and technicians on the field working closely to the farmers. And we are based in more than 40 areas in Morocco, including Laayoune now. As Professor Sraz said earlier, we are putting the farmer at the heart of our concern. And also we are putting science in the heart of our concern too, and our partners. So through this approach, we are reaching now around 400 farmers across Morocco directly, and almost 500,000 indirectly through digital solutions. And the most important area where we are working on with our partners, with the UM6P expert, and also with the Ministry of Agriculture and its local representatives, we are working on soil health. So through our mobile laboratory and also the UM6P laboratory, we achieved more than 150,000 soil analysis and recommendation for farmers. That it's allowed our farmers to have to use the right fertilizer under the 4R stewardship. We are working on water management and also on conservation agriculture, which is tackling the climate change issues. And now we are working closely also with the ministry under our national program, no-till program, which aiming to reach 1 million hectares by 2030. And we reached through Al-Mutmir this year more than 30,000 hectares in different regions in Morocco. The most important thing also is we are working also in animal science and education, because sharing knowledge with our farmer, but also with the student and with the PhD student also, it's very important to share and to spread this knowledge and practical knowledge also with them.
Thank you very much. Well, doctors, professors, thank you so much for sharing with us your knowledge and your experiences. That was very good. شكرا بزاف الحضور ديالكم معنا اليوم. Well, professors, doctors, thank you very much. You have any questions? كاين شي سؤال? Questions? Yes, sir. Can we have a mic for the gentleman, please? We have 3 minutes. On a 3 minutes.
Okay, can you hear me?
Of course we can.
Oui, Sir Rashid, have a question for you. Thank you very much for your presentations. Actually, looking at the data for nitrogen fertilization, so it has been increased 9 times, while the yield only 3 times. So with a ratio 9:3, I want just to have your comment on that. Why we have this, I mean, this non-linear increase with fertilization of nitrogen and yields?
I guess you're referring to into Africa project? Global. Okay, well, and you're saying that nitrogen fertilization is increasing while the yield is not following. Okay. My take on that, I might get wrong, but my understanding of what's going on is what we call nitrogen use efficiency. When you apply nitrogen, of course, you have a response curve, but sometimes you can exceed what's needed in the field. Example, FAO has looked at this very carefully, For instance, in China. In China, if you check on the levels of N in the soils, it's a disaster because they've been over-fertilizing. Now, that's probably an explanation. Now, if you come to Africa, it's completely the opposite. Soils are extremely poor, and there is even no nitrogen to be applied. So I think if you look at the global figures, they are skewed because what we call the global north economy. I talked about China, but if you look at UK, you look at US, you look at most of European soils, they are extremely rich in nitrogen. Actually, for the little story, when I started working nitrogen fixation, we couldn't get the nitrogen to fix nitrogen around the region of Toulouse or Sud-West in France because the soil were rich in N, in nitrate, and it inhibits nitrogen fixation. I hope that explains it. It's basically skewed by the global north.
I think it's 6 p.m. We have to end. So we're sorry for more questions. Any more questions? That'll be the last question, please.
Okay. I have a question.
All right. Yes.
Well, in the UK, a few years ago, they started to get cereals like wheat to fix nitrogen as experiments. And the reason for that, because the main crop in the UK is really cereals, whether it's wheat or barley. So I feel maybe in order to reduce the amount of nitrogen that we add to the soil and finds its way to the rivers and polluting water in the UK in particular. So that was the reason, is trying to minimize the nitrogen that they added to the cereal crops, not to find its way and to pollute our water. I don't know what do you think about really moving with our thinking to also to make the cereals to fix the nitrogen as legumes. Thank you.
Well, I think we need a whole conference to discuss this. But to cut a long story short, this has been a dream for last 50 years or so. It was a dream. Many people have tried. They've thrown so much money. I worked at Arid, as I told you, in the Philippines. Arid has had this program to make the rice fix the nitrogen fixation, at least for about 20 years. If you know J.K. Lada, he spent his life, all his career. Now what? If you want my opinion, I don't think it's happening. Ken Giller, whom we hosted last Science Days at the conference, on who spent his whole life working on nitrogen, does not believe into it. I think it's artifactual. Nitrogen fixation in cereals is still a dream. Will it happen? Maybe I would say in the next, maybe in 20, 30, 40 years from now. But today, it is not doable. There are claims who are not scientifically based. They don't have a strong basis in science. There is always a debate among scientists, but the evidence is really poor for that. That's my take on it.
Thank you very much. So we think we're going to end this. Please, سيد ولي, سيد رئيس الجهة الإقتصادية جوناس المدير، صورة تذكارية مع جميع المتدخلين. تفضلوا. Please, I'm going to ask Madam Fatma, please. Mr. Mark Tester, Rick Dallon, and Edward Bart Leonard, please, for the last picture on stage, if you don't mind. Madam, please. Okay. Outside? Okay, perfect. And we'll be taking another picture outside with all the participants. Come on. All right. So with this beautiful picture, I want to say thank you so much for staying with us the whole day. And there is more to come within the 2 more days coming, 22nd, 21st and 22nd. Merci beaucoup. Shukran. Gracias. مع السلامة. Oh. Micro. S'il vous plaît, n'oubliez pas. Thank you, appreciate it. Thank you. There is a picture outside with all the participants, please, with Mr. Lawali and all dignities. And guests, please don't forget to get outside. And all of you will get the best picture ever. N'oubliez pas de prendre la photo extérieure, s'il vous plaît. Ça va être une super bonne photo avec tous les participants. Nos invités, bien sûr, nos hôtes, en compagnie de Monsieur Lawali, bien sûr, et Monsieur le Président de la région. Merci beaucoup. Et n'oubliez pas de rentrer chez vous. Et bien sûr, demain après-demain, more to come.